All right, so we're 5 minutes late because I'm trying to figure out how the microphone works, right, old guys? And it turns out that this is not actually for the PA system in the room. It is for the live stream. So there are folks who weren't able to join us in person and are instead joining us via Zoom. So I want to welcome all of you who are here in person and also those who have joined the Zoom feed. And I should talk like this, I'm told. So my name is Mike Reed. I'm Dean of the College of Health and Human Performance, and it is my very great honor to welcome all of you to this recognition ceremony for Dr. Tom Clanton. And this acknowledges his service and his recognition as the Billy K. and Betty C. Stevens Endowed Term Professor in Applied Physiology and Kinesiology. I want to start by recognizing the people who made all this happen. Most notably is Michael Spear, our current Director of Communications and Events. This is a special recognition ceremony for a number of reasons, one of which is these ceremonies were Michael Spear's idea. He brought this to our college, he helped us establish this tradition, and we're deeply indebted to him for that. I've seen many of you at our previous ceremonies this year. There have been 3 prior ceremonies, and in each case it's been incredibly impactful, a really precious moment in our college's life, and we owe that to Michael Spear. So Michael, even though you're standing in the hallway hiding, we thank you very much for helping us do this. I also want to recognize Bridget Welch, who you've seen, she's everywhere around here organizing this event. Melissa Cunningham has been very involved in this. Jeremy Clark has been very involved in this. Without the work of those folks, we never could have made this happen. I also want to recognize our faculty, our students, and our staff who've joined us today for this. That's the core of our college. It is who we are. Thank you for being here. This is meant to be us celebrating Dr. Clanton, and I thank you for being present. I also want to thank the special guests that we have, friends, families, and colleagues of Dr. Clanton who've come to celebrate him and to be present for this ceremony. Welcome, all of you. I also especially want to recognize the other endowed professors. Dr. Cunningham, Dr. Valancourt is here. I think Dr. Tucker was unable to join us, but she sent her best regards, Tom. I just want to say a little something. So I see some faces here who have not been present at other events and who may not recognize why we are celebrating an endowed professorship. What's the big deal? Well, rest assured, it's a huge deal. A few numbers. Most of us are scientists and kind of like numbers. We think of college education as a common thing, right? More than 50% of Americans have college degrees or certificates or credentials such as that. On the other hand, even though, again, almost everybody in this room is Dr. somebody, the fact is that PhDs are extremely rare in our culture. Less than 2% of Americans earn a PhD. Among those who earn PhDs, maybe 3 to 5% ever earn the status of a tenured full professor. And among tenured full professors, less than 1% is an estimate for those who attain the status of an endowed professor. It is the ultimate achievement of an academic career. And that is what we celebrate for Tom and for his achievement as the Stevens Endowed Professor. I'm going to turn this microphone over, but another reason that this particular ceremony is special is that we've gotten Dr. Scott Powers to come back and introduce Dr. Clanton. It means a lot to me because he recruited me. He was the head of my search committee. He brought me to this college. Scotty, people have mostly forgiven you for that, given the fact that you also recruited Dr. Clanton and were the head of his search committee. One of the greatest gifts that you've given this college. Thank you for that. So I want to turn the microphone over to Scotty, who will introduce Dr. Clanton.
Well, needless to say, I'm delighted to be home. It's wonderful to see friends and colleagues that I haven't seen in some time, and I'm truly honored to have the opportunity to introduce today's speaker. So I think one of my proudest achievements of spending 33 years on this faculty was the success in recruiting Tom here, and I was really pleased to have the opportunity to be part of that process. So my strategy to recruit Tom was simple. I was recruiting Pam. And so my goal was to convince Tom's lovely wife, Pam, that Gainesville in Florida was a great place to live. So one night we were having dinner and things were going really well, and I was really proud of myself of these recruiting acts, and I was confident that we were going to recruit Pam, and I knew Tom would follow. And Pam mentioned to me that she had an unnatural fear of alligators. So being the quick thinker that I am, I simply relayed that, well, last winter we had this really bad cold spell that essentially eliminated the entire alligator population in Florida, except Albert and Alberta, and they're your friends. So it all worked out. And Pam, and I do hope that you've forgiven me for that small stretch of the alligator population. You know, it's a real challenge to introduce someone that doesn't really need an introduction. But here goes. Tom, before he was Dr. Clanton, earned his bachelor's degree in chemistry. He then went on and earned a PhD in physiology from the University of Nebraska Health Science Center. He did his postdoc at Ohio State, and of course, they recruited him to the faculty. Following 25 years of funded research, very successful research, we were successful in recruiting Tom away, and he joined this faculty in 2007. During his 17 years here, he served this department and college in many ways. He was an extremely successful department chair, and he's mentored very well countless numbers of graduate students and postdoctoral fellows. Most of you know about his outstanding extramural funding record. Most of you know about his many research publications that are highly cited in the literature. But did you know that he once studied control of breathing in chickens? And these are not any chickens. These were leghorn roosters. And if you've ever grown up in the country and encountered one of these can be very aggressive. So Tom, that was heroic biology. So joking aside, Tom's diversity in research is extremely impressive. Over the last 4 decades, he studied the following. He studied the impact of training on inspiratory muscles very successfully. He was a pioneer in studying the consequences of radical production in contracting skeletal muscle. He's published groundbreaking papers, highly cited papers, about the role that hypoxia plays in radical production in skeletal muscle. He studied intermittent hypoxia when hypoxia wasn't cool 25 years ago. His current work on exertional heat stress has produced some cutting-edge studies that he's shown the epigenetic responses in muscle in response to high heat. He's done cutting-edge studies with the impact of sepsis on myopathy and studying the mechanism of that. And finally, do you know that Dr. Clanton is also an accomplished songwriter and musician, and singer, of course? Bob Dylan is fortunate that Dr. Clanton chose science as his day job. Please join me in welcoming our speaker, a speaker of many talents, and my friend for 30 years, Dr. Tom Clanton.
Okay. Thank you all for coming. I think I know everybody here, and it means a lot to me that you're here. And I'm touched. Scotty, I miss you, buddy. And Steve, and I really miss the banter between Scotty and Steve. We used to go to coffee every day, and there was an art form. These guys had a quality about giving each other crap every day about something. It was truly wonderful, and I was always quietly kind of following behind. So hopefully this is working. So this professorship, I've had it for 10 years or so, and so it's not a new thing for me. They just decided to celebrate it, so it's extremely happy for me to have this happen. I've kind of been trying to research BK and Betty Stevens in terms of their history. And BK was one of the founding members of our department back when we were a physical education and health department. And Betty, who I didn't ever meet, I know Scotty had met her several times. I think I've heard she was a firecracker. Does that sound about right? Mm-hmm. She was the dean of women, and she actually had a lot of positions around the country. Interesting family. But they basically, you'll see their name right down the hall, the next room over, their reading room that they donated funds for. And there's scholarships, there's term professorships, there's all kinds of things that they have donated to the college. And I'll be indebted to them the rest of my life in terms of what they have done for us. For those of you who don't know what a professorship does, it's an honor, first of all, but it's also money. Not personal money, but money, startup money, really. Not startup money like a normal startup package, but it's money, the interest that grows off the account, a researcher can use to go and follow new ideas, to listen to one of their students and who has a new idea, and you say, well, you know, I think we could try that. And if you're stuck in a grant cycle, you know, you basically are doing what the grant wants you to do, and to move out of that is very difficult. So this means so much. And I will just quickly say that our department and our college have other ways of supporting us that way that make it possible to do stuff that isn't really easy to do in most other places. So I just quickly, this book, to give you some idea, I found this book. Only copy I could find. It was from the Occidental College. It's still got the number on it in Los Angeles. Teaching physical education in the elementary school. That's what we did at one time. And it's a fascinating thing to look at. I would just want to read you two things. Think about this was written and published in World War II. Here's the introduction. There's a lot to unpack here. I'm not going to unpack it. To the classroom teacher who holds in her hands the power to mold human life. And if you think World War II, there were no female teachers. No male teachers. I'm sorry, I had it backwards. The first chapter, just quickly, Education, a Force for Democratic Living. So, you know, they were going through a time when those were the things that they were thinking about, you know, so it's kind of cool. Anyway, I'm going to give this to the next BK Betty Stevens professor, and so that we can kind of cherish those changed moments. So I'm using this occasion to kind of tell my story as to, I have had a circuitous route through my career. I didn't have a pedigree, really. I know some people get trained by the expert in a field, and then they follow that and they have a history, and I'm not there at all. I sort of found my own path, and it's kind of a long, complicated story, so that's what I'm going to tell you quickly. It's not going to be long, I hope. So this isn't the future, this is the past. And one of the things that has sort of, if I look for threads through my whole life as to what I've been interested in, it really has to do with stress. And what I mean by that is environmental stress, and that's a theme that's carried through my whole career. And it could be internal or external. And I used to only study the short of it, and now I'm kind of into the long of it, so that's where this comes. Our lab is called the Muscle Physiology Stress Lab, so that's where that name comes from. And the stresses I've kind of spent my life worrying about are oxygen or lack thereof, heat, cold, redox stress, osmotic stress, nutrient deficiency, and CO2 and pH. Those variables are sort of the environmental struggles that life had to overcome over the billions of years that life occurred through evolution to actually form us that we can live in this current environment. And so I've basically been studying these same things all my whole career. So this is a picture of where I started, which was as a respiratory therapist. And I hold this in common with Mike Reed. We both started in a clinical situation where we were taking care of this kind of patient. And we got to know them extremely well. We knew their families. And we usually were with them when they died or shortly before that. And they struggle to breathe, they struggle to function, they struggle to even maintain their weight. So all of these same things that I kind of carry through my life, we were worried about these patients, getting them enough oxygen, making sure they could move their chest wall enough to get rid of CO2 and sustain their musculature so they could continue working. And so I took a job, as Scotty said, was in a pulmonary division. I was in internal medicine for 25 years. And it was a perfect fit for me because I had a background as a therapist. And I studied, when I first got there, they wanted me to study chest wall mechanics and muscle fatigue in COPD patients. And that's where we started. We built models of respiratory failure, both in animals and in humans, in normal humans. And we studied that sort of a situation. I'm going to just zip through that. So then I met this guy. So I want to tell this story because it's important history for our department and for us. In the early 1990s, I met Mike and Scotty. And Mike, I credit him with really being the one that convinced me that muscles make reactive oxygen species. He credits other people, but I actually have done electron paramagnetic resonance, and I know. This is the most solid data we have that skeletal muscles during contraction make reactive oxygen species. That was a big thing. He also really demonstrated different probes, different aspects of that phenomena, and it has affected all of our lives in exercise science. He also was, with a group of people, discovered nitric oxide synthase in skeletal muscle fibers, a huge find, and that when muscles contract, they produce nitric oxide. You have to realize that the discovery of nitric oxide, the Nobel Prize wasn't given until 1998, so this was in the early 1990s. I can't tell you how exciting all of this was for us. We were just really excited. We thought maybe, we were onto something, we're going to do something great. And Scotty had an equally important impact. And one of the figures that I use when I talk about reactive oxygen is he was really into enzyme function at that time, aerobic enzymes and oxidative defense. And one of his earlier things he was working on is the antioxidant defense of skeletal muscles, sort of he did sort of a survey of different mechanisms by which skeletal muscles fight reactive oxygen species. And this, to me, was a convincing argument that there was reactive oxygen species being produced, that when he exercised muscles, the catalase activity went down. And that's an indication of reactive oxygen production because catalase takes up electrons and it goes into a state where it's not as active. So this was correct me if I'm wrong, Scotty, but that's my interpretation of that data. Showed a lot of other data. The other related thing that he did a few years after this was to show that inactivity in muscle diaphragm, which is always active, is another form of reactive oxygen species production. So all these things were really exciting to us. And Mike started getting us together. This was at Baylor. When he was at Baylor, he was in a pulmonary division like I was in internal medicine. And as I recall, we went to a Rolling Stones concert and drank a lot of beer and ate Texas barbecue. Those are the things I really remember. It was also a lot of fun. 2 years later, I had that meeting at Ohio State. And then Scotty called me and he said, Clanton, what are you doing? Why are you bringing people in February to Ohio? That's completely crazy. I'd really like you guys to come down here. And Scotty has an international reputation as he and Lou as a gracious hosts. And so we started something which I'm trying to remember how it got started, but it was the Free Radical Underground. And so we met every 2 years, and we were extremely excited. And it was one of the most fun memories that I'll have. I look forward to it every other year. It was such a big event. We brought our students and we shared data. And Scotty, because he had all these connections all across the world, he brought in the top scientists in muscle biology, regardless of whether they worked in free radicals or not, in part to convince them that free radicals are important. But anyway, it was a wonderful time. I think it lasted 10 years or so. Why underground? At that time, you have to realize that the classic muscle biologists didn't believe reactive oxygen species were worth a damn. They didn't think that was anything. There was about maybe 30% that were starting to believe that maybe it meant something. And everybody else, if you go to study section and you listen to what they talk about, they'd say, this is crazy. It's reactive oxygen everywhere. It only lasts microseconds. It's not doing anything, blah, blah, blah. So we were underground. We were radicals, you know. We were behind the scenes, you know, believing in what we were doing. So I was in that realm. I did work in some of the areas, but I think some of the things that I was most excited about, I did at Ohio State. We discovered that I was going to free radical biology meetings, and what I'd hear from other disciplines, almost anything that any kind of change or any kind of stress that a cell sees, it produces reactive oxygen species. And I thought, well, I'm interested in hypoxia, so that was one of the things we did. We had this sort of neat system that we built just to do this, where we had epifluorescence so we could measure oxidation reduction of probes. And we had a very fast system to change the oxygen concentration very rapidly across this muscle. And what we saw was a reactive oxygen signal, which nobody believed, right? It's the same kind of thing. How could hypoxia cause reactive oxygen? It needs oxygen to become reactive oxygen, right? Well, one of the experiments we did to sort of think that that's real was we gave antioxidants. When we gave so when you put a muscle in a really severe hypoxic environment, it loses force. You don't even have to stimulate it. It just starts to lose force because it's building up inorganic phosphate and things from energy problems. But when we gave antioxidants, we got a big increase in force in hypoxia. So that was helpful in terms of trying to convince people that this was important. So again, here we have, you know, too much contraction, we produce reactive oxygen. Too little contraction, we produce reactive oxygen. Too much oxygen, you produce reactive oxygen. Too little oxygen, you produce reactive oxygen. So anyway, somewhere I decided, why don't we try heat? And I'm going to go on heat the rest of the time. That's how I got into heat. So we had muscle, sort of a similar experiment. We used confocal for this, but we basically used a lot of different methods, but we exposed the muscle to heat, kind of heat that muscles see in sort of heat stress, heat stroke. Muscles are about 1 degree centigrade hotter during exercise than the core temperature, so they get hot pretty fast. And sure enough, we saw a huge reactive oxygen signal. When we tried to block with antioxidants, we actually lost force. So it ended up that reactive oxygen and other things related were actually necessary for the muscle to respond to the heat. Long story there, I can't talk about it much, but it got me into heat. So about this time, Scotty calls and says, hey, we got this position down here, and senior faculty, would you be interested? And you kind of heard the story. I wasn't sure, it took a little while. And I thought, well, one of the things that's sort of interesting, first of all, I'm trained as a medical physiologist, not as an exercise physiologist. So here I'm going to go to an exercise department and I'm going to have to teach. Fortunately, they've been very patient with me here. But I thought, how am I going to fit in here? But then I started thinking about, I was doing exercise work with COPD patients and things, so I did have exercise background from that. But one of the things I realized that what we study in exercise science is really how athletes and people overcome these same stresses that life itself, these primordial signals that really drove evolution to make life. In this case, they're really the outer limits of our performance, right? So it was really an easy step for me, and it really excited me that maybe by moving here I might get a creative wave of things to do. So I'm finally getting to what I want to talk about, so I'm sorry. I'm going to take a big jump in time. We did continue to study heat along with a few other things, but largely heat. And the first things that we did, I had like an anesthetized model of heat stroke in mice. We were heating isolated muscle again, looking at cytokine production and regulation. Andy Judge, who I don't think is here, was helping us with some of those experiments. So we did a lot of things, and I'm really proud of what we did then, but we were a little bit lost in the ozone. We didn't really have a good model. We couldn't get money from the NIH. So I knew that the Army supported work in heat. And so I did something really stupid. I put a white paper in to the Army. That's how it works in the Army. You put a white paper in. And a white paper is sort of a short grant, and you describe what you're going to do, what you'd like to do, and are they interested at all. It's a good system, actually. So I put a white grant, and my great idea, I was going to swim mice. I was going to swim them in hot water until they got heat stroke. I thought, well, I have control over their body temperature. It'll be very reproducible, and it's a great idea. So I sent it in, and it gets killed. And these get reviewed internally in the Army. They review, that's who really reviews them, sometimes external, but mostly internal. And Lisa Leone, who was one of the world's experts and a senior scientist at USARIUM, who became a colleague, calls me on the phone and says, "What the hell are you doing, Tom? I mean, why do you think the Army is going to pay you to swim mice? You know, we don't care." And so she says, "I tell you what," and this just was over the phone. She said, "I'll give you a $100,000 contract to build us a heat stroke model in mice with running in like, you know, like soldiers do. You know, that's kind of what she said." So I was really fortunate to have that initial investment, and we've been able to do heat stroke research with this model for about 12 years. We've had continuous funding from the Army. So a big key to that was Michelle King. Many of you might remember her. She's one of my favorite students, one of the hardest working students I ever had in the lab. And she really developed this model by working many, many long hours, trying different ways of running, different ways of training, different ways of heat, different, you know, all kinds of different things that we had to develop. So I was really proud of her for doing that. And it ends up to be the model. Orlando uses this model. The Army uses this model. It's kind of the model that people use because once the animals get heat stroke, they basically have all the characteristics of a human heat stroke. All the same biomarkers show up. They go unconscious. They run until they go into a coma. Sometimes that people consider that cruel, but the truth is 85% of humans who get heat stroke go into a coma. So it's kind of coma and similar very serious neural responses are really the thing that defines heat stroke versus heat exhaustion. So it's an important part. So I had a whole lot of things, a lot of students that worked. We had a great time over those years. But I want to jump to the most recent stuff we're working on. And that starts here with a project that Orlando was the first author on that we all contributed to. But it has become really the key to what we do now. And so I don't want to bore you. This is, I told Pam I was going to talk like this, and she says, "No data." So here I am. Anyway, I don't want you to look at the data. This is how we take glucose into our tissues and how it goes through normal sort of metabolic pathways. And when you do metabolomics, you get different concentrations of these metabolites, and you can tell kind of what's going on and whether there's a blockage somewhere along the line. So we knew right after heat stroke, right during heat stroke, that the animals are in an energy crisis. That was where I was interested. I really wasn't thinking long term. But Lisa, again, said, no, we want to measure metabolomics, and so of the heart, so over a long time period. So we ended up doing intervals of, we collected samples at, we collected mice at different time points after the heat stroke. So they initially have this crisis, but then, within 3 hours even, they're back to normal, basically. 3 hours. The animals look fine. You know, they're already drinking. By the end of the first day, they've already gained back their whole body weight. They lose about 10% of their, 8% of their body weight, and they get it all back right away. They look great. So we're following. I didn't think we'd see anything at all. And when we revealed the data, Orlando remembers this, we didn't see it in the males. We did the males first. There wasn't anything. Well, let's do the females. So we did the females, and look what happened. All these metabolites went to almost zero. I mean, it was amazing. It's like it's blocked. And if you, I'm not going to show you anything else, but the fact is that 9 to 14 days, those hearts were a mess. ATP levels were low, NADH was almost unmeasurable. It was crazy. So we got excited about that, and that has driven the research in the lab since then. So we had the problem that the males didn't show up, right? I mean, the problem is the males are wimps. This is something that Orlando is studying. Males are wimps. We all know this. But the female mice can run twice as fast almost, and they have much better thermoregulation. So it's interesting how that is driven by endocrine things, and that's what Orlando's studying in his new grant. But we had to adjust the model so we get a signal in the males without doing anything crazy. So Christian Garcia, who's a student in the lab for his final project, he adjusted the temperature in the environment, lowering the environmental temperature so the male mice could run longer and be exposed to a longer stimulus. And sure enough, we got the same thing, or very similar to that in the males. So just quickly, one of the main findings is something called a loss of metabolic resilience. It ends up that this phenomena is common to a lot of heart disease. Obesity, aging, heart failure, ischemic heart. And what you see is normally the heart uses fatty acids for metabolism. It's 85% or something of its normal metabolic pathways is coming from fat. All of us sitting here. But if you have a failing heart, you switch over to glucose, ketone bodies, and amino acids, lactate if it's available. And so you get a completely different metabolic scene inside of the heart tissue, and that's what we see at 2 weeks. And as I'll show you in a minute, we get that at 3 months, same thing. So is it true? Is this phenomenon true in humans? At that time, I think we didn't know anything about humans in terms of long-term effects. We knew some people, there's these, anecdotal statements that people have problems after heat stroke, but we didn't really know anything consistent was there. And what we found in the literature was that almost 3 to 4-fold greater incidence in the next 10 to 12 years of heart disease, greater mortality due to heart disease. So we wondered, how does that line up against other risk factors? And if you look in the same time period, the same frame of reference that they were in, the risk of cardiovascular disease with a body mass index of greater than 30 is only about 1.5 times the normal population. Risk of cardiovascular disease with diabetes, type 2 diabetes, is only twofold. So have you ever heard of a cardiologist say, have you ever had heat stroke? They don't know. They don't know that this is actually a risk factor for heart disease. So I'm passionate about this. Somehow we're going to get this across to somebody somewhere that this is important. So real quick, I'm going to try to get through the rest. We decided that we wanted to go longer, and we wanted to give a second stimulus to see if we could pull out what's called a phenotype, which is the structure and functional, something you can measure. And this is a common method to challenge the organism to see if you can pull out that characteristic. So this is work of Jamal Al-Zarani, my student, really did a wonderful job on this, and he worked closely with Ashley Smuter, who did all our heart work on this, and we've ended up working together on the next level. Just quickly, we kind of run our normal experiment, but then after 2 weeks after heat stroke, we put them on a Western diet. So this is what you all are eating. Not really, but it's kind of, it's basically higher fat, higher carbohydrates, and things like that. And then we waited, basically 3 months after the heat stroke, and then took the hearts and looked at metabolomics. And what did we find? Well, first of all, here's the most important data, the weight of the mice. Everybody believes weight. So I told Pam, I told you we weren't data, but this is easy data. So, these down here are the animals on a normal diet. And in terms of, if you look at the overall response, these are control animals that have the same exercise and are handled in a sham way. And these are the animals that had heat stroke. And they got significantly fatter. If they were on a Western diet, that was amplified hugely. And it was in both males and females, but higher in males. So the question we have is this mean that if you get exposed to this heat stroke, does it make you more susceptible to metabolic syndrome and then heart disease, diabetes, everything else that's accompanying that? So just a summary, we get evidence of cardiac hypertrophy with this. We have a 50% loss of plasma proteins. It means that there's probably some kind of endothelial injury that's happening, and how much of that is the weight that we measure, because there's going to be movement of fluid. There is acceleration of fatty liver disease. We know that this diet makes the liver fatty, and that's what we measured, but it's greatly accelerated in the animals that had heat stroke. And finally, the same problem of loss of metabolic resilience in the heart. So we're really excited about that. It really is like important, I think. Where it puts us in, all of us know this in the last 10 years, that we've kind of been exposed, or 20 years, I guess, we've been exposed to the things that we used to worry about, acute problems. We now realize that a lot of individuals that suffer from these problems end up having long-term problems. And we know all of these. You've seen them before. Orlando is studying this one, post-sepsis syndrome. He and the group down the hill, that's a huge thing. But now I think with this work that we're doing in the preclinical model confirms that heat stroke is one of these problems where people have to begin to look for problems much later after exposure. I'm just going to tell you quickly in 2 slides what we're doing now. So we've been studying the epigenetic response. Whenever you see this thing, all of these different problems have been associated with epigenetic changes that can be identified that are kind of predict poor behavior, poor physiologic behavior. So we've been working, I've been working, Jason Brandt is here, and Dr. Claudia, we've been working together to do epigenetics in these animals. 1 month after heat, we studied immune cells, we've studied the heart, when we studied skeletal muscle, and we get large changes in epigenetics. DNA methylation is the main thing we look at. The problem is it's very hard to relate. We also measure transcription. Usually those two go together, but it's very hard to actually match those together in this case. And I'm not sure Jason will agree with this, but I've started to believe that it's sort of like the enzymes responsible for this DNA methylation are like they're letting methylation fairy dust across the genome, and it's just noise. And that's why the animal's not responding well to anything. I mean, there's a lot of noise in the normal response system. So that is I think that represents a maladaptive epigenetic response. And one of the things we're doing now is moving to an adaptive epigenetic response, and that's our new funding cycle. And we're going to try to compare those two as to how they actually work molecularly. Jason and Dr. Claudia are experts at this sort of thing, so we really depend on their help. So we got a new line of funding for another 3 years, so you don't get rid of me quite yet. And we're actually working with humans on the Fort Moore. We're also working with mice, and we're looking at what we're trying to solve is whether we can define an epigenetic signature in immune cells that defines whether a person has long-term heat acclimation or acclimatization. We're using real humans in basic training in the summer before and after they go through 10 weeks of really intense workouts in the heat. And then we're doing the basic things in mice. And Dr. Claudia and Dr. Brandt are with us. Orlando's a co-investigator. We're working in collaboration with Auburn University, who provides the services to collect blood samples in the recruits at these 2 different time points. David DeGroot, who's a PhD physiologist, who is the head of the Heat Center at Fort Moore, and Deb and Finley, who are here and are absolutely necessary for us to do this work. So anyway, that's where we are right now. And just one more thing. First of all, gratitude for this support that's been here for 10 years or so for me. It's made a huge difference in my ability to be creative, and I will be indebted to these people the rest of my life. And this investment in our department and our college is forever. So there will always be a B.K. Betty Stevens professor, and that impact on our college and on medicine and on the things that we do is forever. Just quickly, we're the home of Gatorade, right? So, you know, it's important. Doug Casa is an adopted member of our program. He's the head of the Corey Stringer Institute, which is a big place for heat stroke research in the country, and he's become a friend. Steve Dodd's student, Josh Selsby, has become a good friend of mine, and is doing very similar work in livestock and now mice. And this is an interesting thing as our climate change. He actually exposes pigs to just static temperature of about 98, 99 degrees Fahrenheit overnight, over 24 hours, just static. Well, I'm from Nebraska. We have multiple weeks like that. So for cattle, it's important. He shows structural defects in the heart, 24 hours after that exposure. He hasn't gone long yet, but the point is that heat stress exposure has risks. And I think that's what we're trying to come to grips with, and how much risk there is and in what populations, et cetera. Brady, few of you know too much about Brady's research, but he's done some really wonderful things to serve our community in terms of the potential for heat stroke in our athletic population in Florida. And Brady, where are you? We are #1 still, right? In terms of heat incidences in high school. So huh? Yeah. Yeah, so we're still #1. That's good. I'm sorry, not really. That was a black joke. And just to remind me that that's a very black joke, we have a scholarship in our college for 2 individuals in athletic training. I don't know that I know how to say Erasti, I think, Erasti Outen Scholarship. This young man came to University of Florida and died in like the first 1 or 2 weeks of practice of heat stroke. And his parents donated to our college in his memory. And something I keep going back to think we have this environment here that this is an important topic. So anyway, I want to thank you all very much. Deba, thank you for coming all the way from Europe. Anyway, you know how much I love this place. I've said it many times. It has made my life different. And I have so many good friends, and been a wonderful experience. And thank you very much for being patient with me. Thank you. Thank you, sir.
That was a terrific lecture. Thank you for that. On behalf of the college, we want to present you with this medal. It's engraved in your honor as the B.K. Stevens professor, and we thank you for all you do and what you mean to us.
Thank you, Michael.
Stay here with me. I want to announce that this party is not over. We moved from here to the Dean's Suite in the conference room. We've got a professional caterer there who's got hors d'oeuvres for us, and we're serving both sparkling water in honor of hydration and osmotic stress, but also champagne for those who want a fatty liver. You can work on that. Come be with us. Come shake Dr. Clanton's hand. Come celebrate with us together for a little while longer. When we go down the hall, it's just down the hall and to the left. You'll see that there's a background, a backdrop, for those of you who want to get pictures with friends and with colleagues to celebrate this moment in this space. So, Tom, thank you for this, and thanks to all of you for being here. Please, let's go and celebrate together.